Welcome everyone to this here Exposome webinar series. We have a great speaker today. Before I introduce our speaker today, just a housekeeping. If you need to ask any questions, and there is a Q&A box. When you click on it, it will open a chat box, and please type in your question there if you have any, and we will read those questions at the end of the webinar. Tracy, next slide. Yeah, and so there will be a box where you can type in a question and click on ask. All right, that's for the questions. So today our speaker is Dr. Renrong Wang from Massachusetts Institute of Technology. He's a senior postdoctoral associate in the lab of Professor Connor Coley. They're building several AI models for chemicals. And today, Dr. Wang will talk about deep learning approaches for structural elucidation using tandem mass spectrometry. It's really a great pleasure to have him here. Thanks for accepting our invitation. Renrong, this podium is yours.
All right, thank you. Thank you, Nash, for the kind introduction and also for inviting. Very glad to speak here today, talk about our research on this, talk about our research on deep learning approaches for structural elucidation using tandem mass spectrometry. So I'm Renrong, affiliated with Coley Group at MIT, and very happy to come here today to speak at here. So I think before we really talk about like the technical details about like all the methods we developed so far, I want to still keep like this motivation part as an introduction of why are we interested or feel motivated about this problem. And then we'll dive into two different family of approaches that we developed. One is what we call inverse MSMS models. So in other words, basically spectrum to structure. And the other family of models called forward models, which working from structure to spectrum. And then if we have time, I want to also go through several example demonstrations and successful applications that we have done together, first using our model, and also we also experimentally validated all the predictions made by the model. So I think as Dinesh also mentioned earlier, so I think we should save all the questions to the end, but you should just feel free to type your questions in the Q&A section in the Teams meeting. So hopefully I don't want to convince everybody, considering the audiences of this talk, but I just still want to mention small molecules are important, and analytical chemistry is just essential for understanding human biology, the environment, and also the atmosphere. So there are just many scientific novel discoveries that are just led to the discovery of one single important molecule. Maybe among them, one of my favorite example, and also kind of like relevant to the topic of here, is that on the lower right of this slide, we have this compound called 6-ppd quinone, where scientists found just a trace amount. This compound is going to lead to the death of a special species of salmon. So we have like all these applications and discoveries. So the problem is just how to know the structure of this compound from an unknown. So if we take a look at, let's say, like a 1,000-foot view of workflow for metabolomics, and that's actually basically where our lab was motivated to start looking on this issue. So the experimental and computational workflow is kind of like, I'll say, like established or standard. And usually you'll have a sample of interest, something like a biofluid, plasma, urine, whatever, and just feed it through the LC-MS, which is a combination of liquid chromatography and then tandem mass spectrometry. And then you're going to get a series of mass spectrum. So to this stage, so we assume like we are having separated spectrum for each molecule, assuming there's no like co-elution, whatever. And the next step is just take one spectrum, take one mass spectrum, and run through computational metabolomics tools. And then it's going to help us do the final structural elucidation task, which means that just given one mass spectrum, we want to know what's the corresponding molecular structure of that. So all looks good. I think it's like a pretty standard approach right now. But current metabolite identification workflows are just still falling short. So if you look at the digits from a recent study, current computational methods cannot resolve up to 87% of those tandem mass spectrum in existing databases. Here we're talking about GMPS. So although we believe it's a pretty like promising approach, and there's a lot of value in it, we're still dealing with this so-called metabolite dark matter that are just sitting there. So one of the motivation of us is that let's just try to use deep learning methods like that to help illuminate that space. But I think before we're talking about like all the technical details, I want to just spend a minute in kind of like drafting the high level philosophy of the problem. So we can kind of like start imagining the entire chemical space, including like all the possible small molecules. So that would be a huge space. And when we're doing like structure elucidation to some extent, so we are actually just trying to use spectrum information available on our hand and try to narrow down the space. So usually if you have or like already characterized the spectrum of the molecule that is unknown, so you can usually have you usually have MS1 and MS2, so you could use tools like Sirius or like Buddy that's available there, pretty high accuracy to infer the molecular formula of them. So as long as you know the molecular formula, you can kind of like just narrow down the space to a pretty constrained one. And maybe if you also have the fragmentation pattern, and also like there are some like reference spectrum and structures, so their structure, and also there's a spectrum corresponding to that. So you can use tools or ideas like molecular networking to find those correlated structures to that. So it could further help you to narrow it down using the fragmentation pattern to a very specific subclass. If we just, in this case, look at like the ballpark here of ceramides, so if we're looking at the unknown here, of course, we take a closer look at the space. There are some reference structures with known spectrum that you use to kind of like use the molecular networking to narrow down to the space. But also there are some other structures, let's say, that are in some databases, but we don't know what their spectrum is. But it's still much better, right? We're still kind of like looking at this constrained space, but I would say it's still like pretty huge. And also apart from these structures, I would say arguably we kind of like know them in this space, but there's just many other hypothetical structures that are not in PubChem, not in HMDB. So we don't exactly know what they are. So I think they're more considered like the really novel structures in this case. So in this case, if we want to further narrow it down, it's actually a case study did by our collaborator at Broad Institute, like Clive Klisch. So you could actually use some other clues, like in this case, we have an association of the CPS1 gene, and there's a known enzymatic pathway associated with two molecular known structures. What you might do next is usually we just spot them, right? You could just spot them, and that's what they did, and characterize like whether they are the structures you're looking for. But unfortunately, no. But in this case, when we're validating the structure, we are not only looking at the MS2 information, but also looking at the retention time. So we found that in terms of retention time, none of them is a match. So we have to rule them out. But based on the similarity of the fragmentation pattern, we can have like a pretty high confidence that we're getting very close. So maybe we should just looking at like a more nearby, much narrowed down space. And let's say you could use some like chemical derivatization experiments to further locate the position of this double bond. So I think there are just many stories like this in the domain of like either metabolomics or like just general LC-MS/MS structure elucidation. And we believe structure elucidation as problem is just about how to use the information you have to narrow down the space and give some possible explanations, and finally at the end find the one exact structure you're looking for and be confident and say, hey, like this is the structure we're looking for. So what people have been using in a tool development perspective is that all the methods available for metabolite elucidation. So I want to start with these like non-machine learning methods. I think this is like the standard approach everybody is basically like default is using. So you just retrieval with spectrum standards, and what you're doing is just query a spectrum to all the standards and rank them by similarity. So what I mean is that you can start with an experimental spectrum and assign the precursor molecular formula for that, and that based on that molecular formula, filter down the reference library and only look at the chemical structures with the same molecular formula and compare the experimental spectrum with all the spectrum in the standard library. So if there's a close match, then it's a very high confidence saying that this is the structure we're looking for. So this is, I believe, like a pretty standard approach, and the people have been using that, working fine. But the main problem is that it's just the size of the standard library. So if you look at the best standard library commercially available, we believe it's the NIS23 MSMS with 50,000 unique compounds, which is already a pretty decent amount. But if you compare that to the chemical space of like over 100 million different compounds in PubChem, that's a very, very small portion. So we're not surprised, actually, that current methods just cannot understand or resolve so many MSMS features we're detecting. So what could machine learning or like all our method development effort be helpful here is that we propose there are two different ways of looking and modeling these problems. So one part of it is called forward MS models. And basically what they're working is that we're still doing a retrieval, like what we're doing with like those existing methodologies, but we're retrieving with augmented standards. So in other words, we're developing models that's able to predict additional spectrum from molecules and add them to the standard lab databases. So in such a sense, we can just have like whatever chemical structure in your hypothetical space, and if the model is working fine, it's able to predict spectrum for all of them, and that's going to expand the capability by a lot, right, based on the current retrieval-based pipeline. But there's also another family of methods we developed called inverse MS models. So it's more like tackling the problem directly, which is just, so we call it by inverse prediction. So the input to the model will be the experimental mass spectrum annotated with molecular formula, and the model is going to predict either a molecular fingerprint. You should be able to use that to look up like existing databases, or you can just run like a de novo generation directly and generate the molecular structure end to end. So just to reiterate, so what are the forward models and also inverse models doing here? So the forward model is taking the molecular structure as input, predict spectrum, and inverse models on our definition is taking the mass spectrum as input and output something related to the molecular structure. So the final thing I want to note before we're going to discuss about the technical details is that actually like these problems are not new. If you look back to the 1980s, we found there's a project called Dendro, and it's believed to be one of the first expert AI systems developed by these three gentlemen and their students at Stanford in 1960s. And among them, we have this project specifically called MetaDendro, which is just attempt to infer fragmentation rules from data, the very, very original forward MS model. So you might want to ask, what's the difference right now compared to 60 years ago? Like, are we just if they failed, I mean, to some extent, are we able to fail again? So we believe like there's something different. One is data, and the other is AI and machine learning tools. So on the data side, we're seeing like these are commercially available or open source data sets that with annotated spectrum and unique structures. It's definitely not an entire coverage of the whole chemical space. But if you have any experience of training, like whatever machine learning models, I think you'll very quickly realize these numbers are pretty reasonable size of a data set that you should be able to learn something reasonable if you have like a reasonable coverage of the chemical space. And of course, we have like all the innovations of machine learning and AI architectures and also infrastructures that's going to help us run the models more efficiently. So here's where we are. And that's why we kind of like set out and develop these families of models. So I want to first discuss those inverse MS models, and just to remind everybody again, so inverse models means that you're predicting structure from spectrum, so spectrum to structure models. And yeah, so given the spectrum, those inverse models are going to predict the molecule. And today I'm going to discuss one model we developed for de novo generation. So which basically means that if you have some spectrum detected from experiments, you first, of course, you have to annotate its precursor molecular formula, and also annotate like each of the peak given the molecular formula, and then use that information to generate the generate structures directly. So the basic problem is just like how to do that in the generation step, because we have tools to do all the other steps at a pretty high accuracy. And the problem we propose here, the methodology we propose here is called DiffMS. So it's a combination of diffusion and also transformer-based encoder architecture, and used to do this de novo generation task. So we first have an encoder that's going to take the mass spectrum and also also predict and like an intermediate latent representation. And we have another diffusion-based generator that's going to condition on this latent representation and generate the molecular structure, or basically, technically speaking, the adjacency matrix of the molecule step by step. So it might sound a little bit too abstract for you if you have never heard about or learned about diffusion. So I have another animation for you. So this is basically what happens in one of the examples we're testing. So like at each step, I think it's over about like 500 steps. You're doing the denoising or doing the generation. We just start with a totally random structure, and our model is able to learn how to perturb that in a reasonable fashion and generate the likely molecular structure in such a manner. So that's basically how diffusion, or specifically discrete diffusion, works, and we apply that to this task. And when we're developing this method, DiffMS, the model we have, it's a diffusion generation condition on mass spectrum, and it goes through 3 training steps. So the first step is what we first train a spectrum encoder. So it's like a pre-training step. So given the peak formula, we're going to predict the fingerprint, the molecular fingerprint. And we did this on an MS data set with about like 20K structures. It's basically, in this case, you need the supervision signal. You need to know what the real structure is in order to generate the molecular fingerprint. So that's how we train the spectrum encoder. And then you can do another graph decoder pre-training. So in this case, you are given the molecular fingerprint and predict the molecular structure. And this is where it becomes kind of interesting, because you can basically, in this case, pre-train them on a much larger chemical library. In this case, we use the library with 2.8 million structures, because in this case, we're having pairs of molecular fingerprints and also structures. You can, by theory, generate whatever number you want. And also the final step is just we put these 2 pre-trained encoder and decoder together and plug them in and do an end-to-end fine-tuning. So the final formulation or input-output of our model is that given the peak formula, so in other words, like each peak of the mass spectrum, we first assign a molecular formula to that, right? And then predict entire molecular structure in an end-to-end manner. So maybe another interesting perspective of our architecture is that in the code or software, is that all the training scripts and pre-trained models are made open source, and you can find them on GitHub. So maybe this is another interesting, as I kind of mentioned in the previous slide, an interesting phenomenon we observed. So we saw like a scaling law in the graph decoder pre-training. Let me explain a little bit. So in this case, when we're doing the graph decoder training, we're working on the molecular fingerprints. And as I said, the molecular fingerprints are basically derived from chemical structures. They usually represent certain motifs in the structure. So if the motif exists, the bit is 1. If it's not, the bit is 0. So it's something you can quickly generate if you already know the molecular structure. So in other words, we can run like whatever enumeration or generation of molecules we're interested in, and of course they might be like some weird structures, but by theory we can generate whatever, no matter how many structures we want, and also at the same time we have unlimited number of fingerprint and structure pairs. So this is pretty interesting compared to the value or like the limitation of annotated mass spectrum data sets. Because right now, like if you are looking at, again, the NIS23 library, we only have about 50K unique compounds. So that's still a very, very small portion of the entire chemical space. So in other words, we can basically just use a larger training data for this pre-training step of the decoder. And we found that actually pretty interesting. The accuracy of the de novo generation for mass spectrum scales pretty well with respect to the size of the fingerprint and structure pre-training. So you can see in the plot on the lower right, if you just add more training fingerprints and structure pairs, so it's going to be better and better in terms of the final step accuracy with our different mass model. And we talked about the decoder. So there's another side of it, which is the encoder. And we used another, so the spectrum encoding with we are doing is in this different mass pipeline is using domain-inspired transformers. And these architectures are basically adapted from MIST, which is another paper we published previously at Nature Machine Intelligence. And in this case, our spectrum encoder just introduces several novel or domain-inspired model components. I'm not going to talk about too deep about like all the technical details, but I just want to mention. So we use like, for example, chemical formula representation as inputs, and also encoding pairwise neutral loss fragments in those attention layers, and predicting magma, which is a combinatorial enumeration algorithm that generates fragment structures for the MS2, and use that algorithm to label those substructures. And also we do progressive unfolding for fingerprint prediction, and also utilize simulated spectrum as like a knowledge distillation. We'll talk about that later when we go to the. Sorry, I'm going to talk about like the forward models that we use here to do the simulation later. So if we plug the encoder decoder together, we first train the encoder separately, train the decoder separately, and plug them in, plug them together, and do an end-to-end fine-tuning. So we found DiffMS is able to outperform several baselines on the very challenging task of de novo generation. And the first thing I want to mention is that DiffMS is just better at predicting molecules from mass spectrum over baselines in various metrics. We measured about like the accuracy, which is the exact structure match, MCES, the graph edit distance of any predicted structure compared to the real one, and also the time model distance of the Morgan fingerprint-based structure similarity score. We also use that to measure how close our predicted structures are. You can see DiffMS is pretty consistently outperforming like all the other existing baselines, but also like on a much more challenging data set that requires other distribution generalization. We found all the other de novo generators pretty just struggle, while DiffMS still outperforms compared to them. So that's great because we're in some sense doing like state of the art on these machine learning tasks, and also they seems to have like pretty broad applications in various domains. But if you just look at the numbers, we have to admit that they're still low, and we're not saying like we can safely trust them in any of those like existing projects. So we kind of like try to ask why de novo generation is so challenging. So we looked at several successful and failure cases we had here. So DiffMS sometimes actually correctly predicts the scaffold, but fails to assign the exact structure. So if you look at the failure case here, you can see we are actually getting the scaffold correct. But the main challenge or the mistake we made here is that we put the hydroxyl group on a wrong position. And you can kind of like argue you sometimes are not able to distinguish them between some of those like isomers because some of the cell bonds will just not fragment usually. Like such a phenomenon is also pretty interesting or You can see here in this example on the lower side of the slide where we have leucine, isoleucine, which is just a slight difference on the carbon side chain. But if you characterize them, you frag them, feed them into ESI MSMS, they just give you almost the same spectrum. So in other words, ESI MSMS itself seems not enough to telling some certain isomers in many cases. So it seems, in other words, it seems pretty challenging if you want to have like perfect top line accuracy. But of course, I think we still have a large room of improvement in that case. So that kind of like introduces why are we developing this family of forward MS models. So we're going to the results later, but I can tell you like they seem to be much more reliable and performs better on several benchmarks, and they are more recommended for immediate applications right now. So these family of forward MS models, as I said, is applying or like performing from structure to spectrum. So given a molecular structure, we're trying to generate the molecular spectrum. And what forward model works is that it's going to mimic what's happening in the mass spectrometer. So just in parallel with the like the physical process, you have something coming from your column, right? A droplet, and you have some molecules in it, and it gets ionized and goes through the collision cell, fragmenting into small pieces, and finally detected on the mass spectrum by its MZ value and intensity. So just in parallel to the physical process, we developed in parallel a learned simulator. So the simulator is taking the precursor molecule as input and predicting the MZ values and also intensities as the predicted spectrum. So we covered that previously, but I want to mention it again. So you could basically build elucidation pipeline with forward models by augmenting the standard library. So in any way, you have kind of like the coverage at least of everything in PubChem. And if you're able to enumerate any novel things, you can also use like an existing forward model to mimic them, to simulate them, and also rank like all the proposed or all the novel candidates you're proposing. So when it comes to the method development of these forward models, we found spectrum representation is kind of like the key question. So that's like the key insight when we were developing these family of models. Because of course we're not the first one to come up with this idea, and the people have developed like let's say the Bain prediction models. So these models, let me explain a little bit. They just use like a fixed length vector to represent the like the entire mass spectrum. So in other words, if you have let's say like a mass band of 1, so everything within like for example like 50 to 51 is merged together, all the intensities are merged together onto that one digit. So of course I think it has benefits. It has benefits, like it's data driven, pretty good, and also it's fast and simple because you only need to some extent you can say like you only need like an MLP layer to do the mapping from the molecular representation to the spectrum. But the main drawback is that they have inaccurate masses because you have no constraint on like where you can have an intensity, where you cannot, and also there's no physical constraint, so you can just generate or predict some unreasonable peaks at an unreasonable MZ. So people have tried to kind of like extend or like improve that pipeline. So we have like these families of models called formula prediction. So despite just predicting like a fixed length vector to represent the spectrum, we are first predicting the subformula of the precursor formula, and then use our model again to predict intensities for each of the subformula. So it gives us like a more physically constrained space, because basically we're right now generating like all the possible subformula. So that's a much more reasonable and constrained space. But we're still losing the substructure information, which is making the methodology, we believe, now perfect. But there's actually one like a one family of method that's being very close to the physical process, which is fragmentation prediction. So these family of models, or I think I should say they are algorithms. And they just try to enumerate all the possible ways of breaking the precursor molecule and generate like all the sub fragments of the previous one. So it's pretty interpretable because you know how every fragment comes, and also it's physically constrained. But it's slow because you have to run this full enumeration. And currently it's heuristic. So in other words, where people are just using like some traditional algorithm, heuristic scores, to decide which fragment to keep in the final prediction. So by being motivated by all of these advances, we thought an ideal model in this case should first use fragmentation and learn from data and be able to scale to train on thousands of examples and also avoid exhaustively fragmenting the molecule. So here it comes to our method called iceberg. And iceberg learns to fragment the molecules in a deep learning based manner. And if you compare like the fragmentation graph I show here, so current methods kind of like require a complete enumeration, giving us like this pretty dense fragmentation graph, which is pretty computationally intensive. And we can have like instead iceberg simulate the graph. So in such a sense, we can only look at, so we can use learning to only learn like what are the important fragments and only generate them and read them. And based on that, the key insight we're having here is that we should develop a two-step model where the model one learns to generate only the important fragments, so giving us this like much pruned fragmentation graph, and also the model two learns to score the fragments to predict the final spectrum. And a little bit detail about how it works is that so at each step, when you're parameterizing, you should parameterizing the candidate generation by atom removal. At each step, the model predicts whether each atom should be removed to generate the subsequent fragments, so that you can have this iceberg-generated fragmentation tree. And also, you were using like graph neural networks and MLP layers at each step to predict like whether this atom should be removed in the next fragmentation step. And based on that, so we can generate in an autoregressive manner a fragmentation tree. And we have another iceberg model two, basically a set transformer architecture that learns to predict intensities at each fragment to maximize the overall cosine similarity to the true spectrum. So if we apply these so basically, yeah, we build this model, train it, test it, and we found that iceberg accurately predicts fragmentation patterns, in this case on the NIST-20 dataset. And we apply two different kind of splits. One is more like an in-distribution split, we call it random split, and the other is more assessing like the auto distribution generalization ability. We use the scaffold split. And if we ask the question of just first, how well do the predicted spectrum match those experimental spectrum? You can see from the bar plots, iceberg, like the latest method we have right here is performing pretty well compared to all the other forward prediction models. And also, if we ask the other question about like how well can we retrieve the true structure from a list of candidates, so this is what's shown here on these curves, on these two curves. We're also performing pretty well and a pretty large improvement over all the existing method. So if we take a closer look at these curves, again, like we are trying to answer the question of just how well can we retrieve the true structure from a list of candidates, because I believe that's like the most common application scenario you should expect of using these family of models. So we first, I should say, we significantly improved the retrieval accuracy with a 40% top 1, 92% top 10, and the previous CDVR was only 25% top 1. And also, we almost doubled the accuracy from several widely used traditional methods like MATFRAG or CFMID. And also, the accuracy dropped from in-distribution split to the outer distribution, the scaffold split, is, I think it's pretty acceptable. So we can see a drop in top 1, which is not too surprising. But if you look at until top 10, it's still like 92. So pretty consistent and also not that bad when you're assessing like the outer distribution generalizability. Yeah, if you have like a stereotype of deep learning models, I think it's pretty robust and consistent. And so all these together, the high accuracy on the benchmarks, although the robustness, gave us the confidence to pursue several real-world case studies we're going to talk about shortly later. But before that, there's another, I think, pretty interesting and important evaluation we did. So we also compare iceberg to the performance of Sirius, which I believe is like the default methodology everybody is using right now for structure ID. So we compare iceberg with Sirius on several new spectrum. I'm saying that because comparing to Sirius used to be a little bit tricky actually, because I'm not confirmed by anyway, but I'm over 95% sure about that Sirius was just trained on whatever spectrum available, which is, I think it's reasonable as a tool that's meant to like help everybody to get the structure from all the unknowns, right? But it makes it a little bit tricky to compare that fairly if you want to do a benchmark. So in this case, we're pretty lucky. We have this recently released library called MSNlib. So we just took that library and keep like average spectrum in that library that doesn't exist in NIST, doesn't exist in GPS, and we have I think a subset over 10,000 structures, and we use iceberg or Sirius, all the other methods to do predictions on that. So you can see here iceberg performs pretty well and a very large marginal improvement compared to all the Sirius methodologies. And I want to mention, so there are like different configuration series you can select in the software, and the default setting is bow only, so we're still better than that, especially if you look at like the top 2 to top 10 accuracy. But I want to mention, so iceberg is looking up the entire PubChem library in this evaluation. And if you just ask Sirius to do the same, look at the entire PubChem library, you're going to see like a much larger head-to-head comparison. If you take it in that way, it's like over double the accuracy of the Sirius PubChem retrieval. So the one more benefit of iceberg is that it's able to predict several chemically interpretable predictions. So in other words, for each of the peak, of course, we predict its intensities, its MZ value, but at the same time, we know what are the chemical structures or the chemical substructures we're predicting. So giving us the ability to come up with what are we doing here. So this ability is especially, again, brought by the two-step model design, where we have like the first stage model that's going to give us like this, as I said, like the fragmentation graph, and also the second model that's going to score each of them. In other words, like predict the intensities in a regression manner. So I think we still have like 10 minutes or so. So I want to quickly go through several example demonstrations of successful applications. So just in this case, we just took iceberg and teamed up with several domain scientists working on metabolomics and also other scientific fields, and see if we can have some interesting or novel discoveries based on that. So in this case, we worked with our folks, Dr. Claire Klisch and Dr. Fulian at Broad Institute, and we use iceberg to identify 3 novel biomarkers from clinical studies. So this is one from nurse health study, and we're looking at, I think, a pretty typical setting of those clinical studies, where we first start with a clinical cohort, a group of volunteers, split them into group A control, group B disease. So in this case, they're person with mental depression and PTSD syndrome. So in this case, we took their biofluid into its plasma and run through LC-MS. And since they have different phenotypes, I think we should expect there should be something different in their biomarkers, right, in their metabolites. So we just ran the statistical analysis and identify like what are the differences or like discrepancies between them. So we use a p less than 0.05 criteria and filter down all the known or unknown metabolites. So in this case, we have a list of unknown biomarkers of interest in this case. So in other words, the setting we're having here is that we know it's an important compound, and we have its MS. So we want to know what's the molecular structure corresponding to that. And on one of the examples, so we just look at one unknown of interest with these criteria or profile, and it have a p equal to 0.014. And iceberg is able to predict this compound. It's called hercycline. And we were able to also experimentally verify this is also the structure we're looking for. So in this case, I think, sorry. So what we did in this case is that we first used iceberg to rank like everything with the same chemical formula from PubChem, and we just look through them, look all the rankings manually, and decide like which one we want to go after. So in this case, I think the iceberg top prediction seemed to be pretty promising. So we just bought it. We bought the authentic standard of it and run through the same LC-MS instrument. So we can see that under the experimental validation, we'll see a pretty nice alignment between retention time and also a nice mirror plot with matched peaks in the fragmentation pattern. So all these evidences, I think, together are pretty strong evidence to say like hercycline, this compound is just the one we're looking for in this case. And I think another interesting stuff I want to mention here is that, so if you look at like all the peaks we plot here on the upper right of the slide. So you can see here on the first row we have iceberg predict the intensities at different collision energies and also the intensities of the standards, the reference standard we got at different collision energies, and also finally the plasma sample we got, and of course like their intensities at different collision energies. So you can see here, so although there you can observe there are some discrepancy or some error or whatever noise you can observe in these fragmentation patterns, either predict or experimentally characterized, but you can see a pretty nice shift from lower collision energy to higher collision energy. Also, even our model was able to capture that. We didn't infer that in any way, but I think it learned that automatically from the data, where at a lower collision energy, you should have like a much stronger precursor peak. And when you gradually increase the collision energy, you're getting like more and more fragments in the lower MZ regime because, of course, the molecule is fragmented more thoroughly. So we believe that's like a pretty interesting case. So we had two more successful cases. One is from the same mental depression study, and we were able to identify successfully this phospholipid as a biomarker. So it's ranked at top 2 among like all the PubChem structures by iceberg. And of course, so we also get its authentic standard and run through the same instrument again, have a nice alignment between retention time and also MS-MS fragments. But also, sorry, on the last case, we have like this this compound, this unknown associated with tuberculous meningitis. So we also use iceberg to rank everything with the same chemical formula. We predict this one at top 1. It's a combination of GABA and arginine. We bought it as someone to synthesize it, bought it, and run through the same instrument again and see, and I mean, observe like a nice match on the retention time and also on the fragmentation pattern. So I think this is just a kind of like a very high level introduction what we could do, or like basically what iceberg could do to enable like novel scientific discoveries. And we might not have time to go through all of them today, but we also used this tool to enable several other new paradigms, let's say in environmental science, where we identify a novel hydrolysis product for the pesticide. So in this case, they're having, like, as you can see on the upper left, they have different kinetics when you put in the pesticide starting material with like either sunlight or water, or if you just put in a dark sample and water. So when you have sunlight, it gets like made at the very beginning, but consumed afterwards. But if you don't have access to sunlight, only water, it will keep accumulating. So I think a pretty interesting kinetics, and we were able to identify the identity of this degradation product. And also another interesting thing in synthetic chemistry is that so people are usually like doing experimental screening when they're designing experiments, designing reactions. So one of the main bottleneck here is that you have to run the experiments manually, usually one by one. So that's a pretty limiting factor, and we found actually you could use iceberg to help you to deconvolute all the products you're making here, even though they have the same MZ. I mean, precursor MZ. So like basically a traditional method using only MS1 is not able to tell them apart. But if you have iceberg to predict like all the possible reaction products and also how they will fragment, you should be able to easily tell them apart with such a methodology. And finally, there's another interesting, I think, case study we did. So we identified several intermediate products in a biosynthetic pathway of this plant called Vicinium somnifera. So it's just like starting with 24-methylene cholesterol, synthesizing several and ending up at several like bioactive compounds, and we were able to identify or annotate several important intermediate products. So I'll skip this part about the details and jump to the summary. So I think the story or like the main technical thing I want to talk about is just computational methods and how they can help navigate over the dark matter of metabolites and also everything related to LC-MS and also the vast chemical space. So on the modeling side, so we first talk about those inverse models, such as different mass, and they can determine the structure identity given spectrum information. Well, we also have a family of forward models. We discussed the iceberg. I think it's pretty much like the state of the art right now and works pretty robustly. And it can simulate fragmentation to determine the spectrum given some chemical structure. So yeah, by the way, so all these methods, they are open source, so you can access them on GitHub. Yeah, so that's like the model development part. But if you try to apply that to any existing campaigns, so we say that with the knowledge of a chemical space or with the knowledge of the candidate space, basically, one can employ forward models to predict the candidate spectrum for comparison and rank against the spectrum of interest. So in such a way, we built these family of models and also had these interesting case studies, and we experimentally validated those successful case studies. And what's next? So one thing we've been thinking about is just how can we trust the structural annotations made by models? Because you can see, at least, I mean, if you look at the iceberg, I think it's performing pretty well to some extent. But if you look at the top one on a more like a controlled setting of NIST, it's still like 40%. So over half of the time, if you only look at the top one, it's making still a lot of mistakes. But of course, like there are several, I think, it could be tackled in many different ways. And also we should not just blame the model on that, because there's also some, like, as I said, like some structures you cannot distinguish them from LC-MS. But I think what's important here is just to build structure-aware confidence estimation models. That's what we are doing right now, so that you can annotate like what subgraph of the structure is where you are confident. And maybe let's say like I'm not confident about this hydroxyl group, maybe it's fine, but I think we should make it clear to our users, and that will be, I think, very helpful in expanding the applicability of the model. But also, we are also actively thinking about how are we going to deal with those truly unknown and novel structures. Because basically, as you can see from the experiments, I think different mass or like any other de novo generation models, they're still falling short in terms of the absolute accuracy, which is not surprising because it's a much like unconstrained space and also much more challenging. But how are we going to further improve them, and hopefully like to an extent where the accuracy becomes pretty acceptable and we could use them and trust them more broadly. So with that, I want to say thank you. Thank you everyone for coming here today. Thank you for the attention, and special thanks to the Here's webinar. Very great opportunity for me to present in here. But I think I should still give credit to the entire MIT Coding group, and also MLPDS at DSO giving us the funding support. And I think especially we have like this MS sub team, or like in the subgroup in the Coley group, where Professor Connor Coley, of course, gave us like all the guidance and helped us sponsor like all the navigate through like all the dark matter of research, but also Mernali and Sam, who developed together like all the methodologies, and also Bo and Jules, who helped carry out several of those important experiments. They are the real scientists here, but also our collaborators at Broad Institute, Clary, Julian, and Northeastern University, Jinko, Naring, David. I think they're also the folks who carry out some of the experiments. also like other students who helped a lot, Magdalena, Monty, Ray, Grayson, Azal, Jenny, Jai, and also our collaborators, great collaborators, and UC Riverside, May, Michael, and Reza. With that, I want to thank everybody, and I'm happy to take any questions.
And Ron, that is a fantastic webinar on this very important topic. We are open for questions. You can type in your question. We can also unmute.
Yeah, thank you.
Yeah, great presentation. Really promising results. I think just great additional work to trying to achieve the holy grail that we have for mass spectrometry and being able to definitively determine chemical structure of things that we detect. So your point on one of your future work points, which is in the area of false discovery rate, assessing that, it's really an important point. And I wonder if you've considered bringing in semi-orthogonal data types like collision cross-section, as can be measured with ion mobility, since a lot of instruments these days do have an ion mobility component. Have you thought about that? And then a second question is maybe more philosophical in whether a tandem mass spectra is specific enough, in terms of discerning between chemical structures. I don't know if I expect an answer, but I think that your team perhaps could explore that question on, whether what can we truly expect from a tandem mass spec ever? Would it ever lead us to a one-to-one match, a top hit? So that's more maybe philosophical question.
Yeah, thank you, Tom. I think they're just very, very wonderful questions. We think about that all the time. And for the first one, I believe there's a lot of opportunity. As you said, you can predict like the mobility, for example. And also, I think another thing we've been thinking about is that you can actually get other kind of spectrum, like GCMS or something else that's hopefully going to give you some different fragmentation pattern, but also still easy to run in a sense that you don't have to separate the sample into, you don't have to purify the sample. So I think that's like the benefit we want to preserve. Right, but again, like if you are able to purify, then maybe like using NMR, I think it's still like another interesting or promising thing. So of course, I think working on like different models in, let's say, like analytical chemistry, I think that's a very, very important perspective. But I think our team here right now, I think we still want to focus on the LCMS first. Because I think we will be curious, like at least just how well can we do, and also when are we going to hit like the upper limits of the I mean in some sense like the physical or instrumental limits of all these instruments. So for our second question, I think based on my understanding, I think there's just definitely more people are more knowledgeable about that in the audiences than I am. But based on what I learned, I believe the answer is no. I think we cannot, in many cases, we cannot narrow down to like one exact structure using only SMS. And well, in a sense you could, but you have to like I mean you have to like synthesize it and get it back, right? Which is take you like several $1,000s and maybe like several months to do it in one run. So yeah, technically you could, but I'm saying like if you just don't want to deal with anything like that, just predict it purely in silico, I believe there's still several limitations. So we want to do that. And again, I think, so just structural elucidation is all about how to narrow down the chemical space. And I wish like what we are doing right now is going to be helpful at least to some extent to everybody who are just struggling with these challenges.
Thank you.
Thanks, Tom.
So I have a question. Do you notice the accuracy differ by chemical classes? And underrepresentation of some chemical classes that we can improve in the spectral databases.
Yeah, that's a very good point. So I don't have the plot here, but yeah, we do have one. So you could see some differences in different chemical classes. We didn't look too deep into like the real reason of why there's like an uneven distribution, but I believe like if it's easy to learn or whatever, or like more examples in the training data set, something like that. I think those are probably the main reasons.
Great. Any other question from audience? If not, thank you, Renrong, for this excellent webinar. Yunjia, you want to ask? 1 second.
Hi, can you hear me?
Yes.
Hi, I really like your approach, like sort of the idea of simulating, I believe in this talk is collision-induced dissociation via the deep learning to generate fragments. So can you elaborate a little bit on how the model accounts for instrument-specific parameters like collision energy, polarity, etc.? And what is your view as the major remaining gap in terms of modeling real-world spectral variability?
Yeah, very good point. So if we talk about iceberg, which is I think like what we recommend, if you're thinking about we just apply that to a existing application, I think that's what we're talking about. So I think the iceberg stuff, we have a pretty wide coverage of different ether types, either positive or negative. So polarity issues, and also we just learned how to handle different like collision energies. So that's also incorporated or baked in our model. And I think so far all the evidence we have seems that it learned it pretty well compared to like different collision energies. So any other discrepancies, I would say I'm afraid we're not either aware or like explicitly handling them. But if there's any like very specific thing you believe that are important, I think we're more than happy to also look at them and see if we can incorporate them in the future.
Yeah, thank you very much. Great talk.
Tracy, can you
Congrats.
share that slide? So we will have the next webinar on chemical mixtures and their impact on children's health on December 16 at 2 p.m. by Dr. Elena Colicino from Mount Sinai. There will be an email sent out to everybody for registration. Thank you, everybody, for joining. Thank you, Dr. Wong, for this great webinar. Have a great day.